Must have been too exciting. You slept well. No. These are students.
There's just a little break, probably. Sorry. They don't. They each have 5. It's just that we had thought we'd have more presenters in that period. We can start at 8:20 instead if we'd like.
No, if you just give me.
How about I just be mile mark with 1 minute on the time?
Yeah, as long as you get that one done by 9, you get 10 minutes for each. I think it's just we're just talk more.
Oh, yeah, it's very good. Oh. So we have a little bit of style when we go birding, but do another
That plus the calipers, I didn't see that. It looks like it. Looks like it.
Those calipers were connected for that. Love you.
Sure.
It's okay, it's better light. Good?
I don't time, but I wonder if it's all ready to go. Morning, we have a round of short, rapid talks. And our first one is Jessica Boobert from Illinois, who's going to talk about the deployment of high-throughput plant height mapping systems on GF germplasm. She's also known as the Midwest expert on broomcorn.
So glad the bar is used. All right, good morning. This morning I'm going to talk a little bit about the development of a high throughput plant height mapping system that we used on Genomes to field germplasm. And so this was a collaboration with Huanjian and Liu Jun from the Department of Agricultural and Biological Engineering at U of I. And so we've talked a lot about the different platforms for phenotyping, so I'm not going to really touch on that because of time. But I will say that we decided to use a UAV-based system because it was high throughput, it was pretty affordable, it wasn't reliant on terrain conditions, which this year we had a lot of mud, so that was great. And the plot numbers we could do were only limited by battery life and the number of batteries that we had. So this is the UAV system for any of you engineers that are out there that know what this means. It was developed on a DJI Matrix 100 platform, which has a DJI manifold computer built into it and an N1 onboard flight controller. The RP LiDAR A2 sensor was hardwired to the manifold, and then there's an internal inertial measurement unit and a DJI ACE-1 GPS receiver that are both attached to the controller. And then that is wirelessly attached to a autopilot system from called AgVision, which is from Ag Census, and that's a startup company out of Champaign. And that whole system is run off a smartphone. So the data processing was the biggest part of this project, and so I'll just go over it really quickly. So the GPS data and the LiDAR sensor data was both collected on the onboard computer, and then once the flight was over, it was imported into MATLAB, where the GPS data was calibrated and fused together based on the flight information from the IMU. And then also in MATLAB, the scanned points from the LiDAR were calculated and calibrated based on the angle of the LiDAR and then the rotation and the tilt and the height of the UAV. Then the ground height was calculated and a dense point cloud of plant heights was created for the entire field. And then from that, we went back to GPS data that could relate that point cloud back to our plots. In our case, it was the GPS data from the RTK on the planter. And then the heights were extracted in ArcGIS. And then just an overview for measuring plant heights manually that we used for ground truthing. I did this with a bar-coded adjustable height stick and a handheld scanner. We measure plant height to the tip of the tassel. For parts of the field, we measure to the flag leaf ligule, and then we do ear height to the uppermost ear-producing node. So once they had the processing down, we did some field testing. And this is the field we tested on. It was 2.5 acres. It took 8.5 minutes to do the flight over this field. As you can see, this is our nitrogen nursery. So the genomes to field subset of the genomes to field's germplasm is in this field, as well as a variety of other inbreds and hybrids at high and low nitrogen. And you can see the flight parameters if you care. So this is on the bottom left, you can see the really dense point cloud that was generated from the flight. So these are all the scanned points. And then on the right of that is the actual map of the heights that was generated downstream once we compared that to the RTK off the planter and gave those heights a plot designation. And then this is just the manual heights that they were compared to. And if you look at that picture, you can see the high and low nitrogen comparison that we have in this field. And so I'll come back and touch on that later. So these are just some really preliminary results. This is before we gave them any feedback and then had them come back to us with some adjustments. And so on the top left, you can see the overlay of the manual and LiDAR measurements. And they overlay pretty well across the field. What's really good is that you can see the third quarter of that is where the inbreds were planted, and those are shorter, which is what we would expect. And then on the right is where we regress those measurements against each other, and they have an R-squared value of 0.87, which is really exciting. And then on the bottom, we're just showing he went out and did these flights, 3 flights between August 2nd and September 17th. And you can see that the R-squared values are really consistent, as are the rest of the error terms that he looked at. And so that means that there's a really big window that we can be taking these measurements in with this platform and still getting consistent results. So that would be good news if we wanted to deploy the system over multiple locations where it would take a while to collect all of the data. So we started with plant heights, I like to say, because I don't like walking around in a field with a stick and a bunch of spiders. Other things I don't enjoy doing is standing under a wood chipper catching high velocity stover projectiles. So the next thing we're going to look at is if we can relate the hyperspectral images that we took to the nitrogen response in our paired plots. So we're going to look into that. And then the last thing I want to touch on is just a little bit of an overview, a high-level overview of what sort of I'm going to try to do with this G2F data, just in the spirit of an open community. If anyone has any suggestions or comments or ideas or is already doing it, please come talk to me after the talk. But the idea is that we're taking all this technology and we're trying to integrate it with our phenotyping, and then we're collecting all of the weather data and the soil tests and the genotyping, and we're trying to make predictive models, and we're trying to make those predictions across locations or across different years, so just different environments. And the idea here is that we are collecting, at our site, we're collecting the same information, the same phenotyping information, we have the weather data, all of that, but we're growing a subset of those genotypes under intensively managed high and low nitrogen plots. And so the idea here is that we're hoping that by taking this wider calibration for nitrogen use, we'll be able to improve the training on nitrogen modules. And that way, when we bring back in all of the information that we have from your locations that have a soil test with soil N and also the agronomically optimum nitrogen that was applied, we'll be able to improve our predictions not only across locations and years, but also across any nitrogen rates that people might be using. So with that, I'd be happy to take any questions, or I can talk to you afterwards if you want to talk about the last couple slides. Thanks. Oh boy.
On your nitrogen experiment, I think that's a great idea. Does it matter what form the nitrogen's in?
Yes and no. I mean, we break it all down to the amount of nitrogen, just straight nitrogen that was applied, so we can do that no matter what type of nitrogen you used. Obviously, it would be a better experiment if the same type was used everywhere, but depending on if you're using the same field year after year, you're rotating, we can look at the soil test and see how much that nitrogen was actually available in the soil. Also, if you did fall or spring soil testing. So we have ways of sort of accounting for that, just because there's no way to have everybody do the same.
Nick.
I noticed for your
nitrogen test, you have a pair of ears that have a marker
Oh.
for high low nitrogen. Can you tell us about the sensor you built for that?
Yeah, so that's the red fluorescent protein on a zein promoter, and so the idea is that the more starch there is, the more red they'll be, because it's a storage protein. And so we use that to measure the amount of starch or protein in the ear, which gives us an idea of kernel nitrogen or grain nitrogen.
Wonderful, Jessica.
Thanks.
That was a lot of information in a short talk, expertly put together. Yeah, take that, thanks. Mike Baseggio, will you come up, please, and tell us about improving vitamin levels for human health in sweet corn from Cornell? Would you clip that on, please?
Thank you. Good morning, everyone. I am Matt. I'm going to be presenting on my project on nutritional genomics for sweet corn. Carotenoids and tocochromonols are compounds produced by plants, and they have several health benefits. As antioxidants, they may prevent cardiovascular disease and specific cancers. Also, certain carotenoids, they may reduce the risk of age-related macular degeneration, which is the leading cause of vision loss for people over the age of 60. However, certain segments of the population still do not obtain the necessary amounts of these compounds. So my project is trying to understand the genetic mechanism of the natural accumulation of vitamins in sweet corn kernels, and in order to convert locally adapted sweet corn lines into nutrient-dense lines. In order to achieve that, we use a diversity panel with about 400 inbred lines. We harvest the sweet corn at fresh stage, and then we flash frozen the kernels. We ground, and then we analyze carotenoids and tocochromonols using HPLC. And the panel was also genotyped using GBS. We conducted a GWAS, and we found some associations with known candidate biosynthetic genes in those pathways. For tocochromonols, we found an association of VT4 with the alpha tocopherol, which is it has the highest vitamin E activity. And also we found associations with VTE1, another gene from the tocochromonol pathway. For carotenoids, we found associations with CRB1 and beta-carotene, which is the main provitamin A compound, and LIKE, which is also another gene from the pathway. So these genes were already reported to be associated with those traits for field corn. Something particular of this panel that was not observed for dent corn, for example, is that we observed associations with two kernel-type genes, which are the shrunken 2 and sugary 1. As you can see in the pictures, this is the two different types of sweet corn that we had in the panel. And so this might suggest that these genes could have an effect on the accumulation of tocotrienols, which are also accumulated in the endosperm. For tocotrienols, we also found association with this gene, HGGT1, which is responsible for producing a precursor for tocotrienols. And however, when we assess the SNP at that position, we can see that for shrunken 2 lines, for example, most of them have the favored allele. So because of this confound effect, we are not able to make sure that the effect seen between the two kernel types is because of the mutation for the starch gene, or is just because of this effect that some type, the shrunken 2 doesn't have both alleles. Other significant markers are not close to any candidate biosynthetic gene, but they were observed in this other study using the NAM population. We can see here the joint linkage interval, confidence intervals, and some of them intersect with our hits, particularly on chromosome 1, 4, and 9. So we are now checking those locations to see if we find any other candidate gene. For the breeding program, we use also a whole genome prediction using RR-BLUP. So we calculated genome breeding values, and then we ranked the lines. So we selected the best lines for the traits of interest, and then we separate them by kernel type for sugary 1, shrunken 2, and then sugar enhanced. And then we use the results from GWAS, and then we assess the haplotypes of those locations that we found hits for GWAS, and then we made the crosses with complementary disfavor allele for these lines, and then we cross the line 24, 25 to those lines. We have made over 150 crosses in the past 2 years, and this is one of those. And this is gonna, these crosses are gonna be, gonna compose a breeding population for nutritional quality in sweet corn. Besides the HPLC data for carotenoids and tocochromonols, we have also extensive information about ear traits. So we have been measuring ear size, weight, and color. We also conducted a kernel ionomics study. So we have information particularly about iron and zinc. So we have all these other elements that we could use to increase nutrients in sweet corn. And for the past few years, we have been collecting field-based high-throughput phenotyping data using both an UAV and the ground-based phenome, which collects several measurements, including NDVI, kernel temperature, and plant height. As for the future, we plan to use AMP-seq to further characterize the haplotypes and the key positions. We are also collaborating with Bill Tracy, Marcio Rezende, and Michael Cantor to generate a de novo draft genome for sweet corn. And we even have been talking about having a G2F for sweet corn. So thank you for your attention, and I'm happy to answer any questions that you may have.
So the fact that the tocopherols co-localize with sugary and shrunken, to me doesn't seem too surprising, because when you have smaller kernels, you're going to have more of those compounds, right? When you have less starch. I mean, is that adjusted for the...
So the question is that because of the different size of kernels, that if that was adjusted, and that's not the problem that we are seeing, that's why the association. So we measure the fresh stage, so the kernels have similar. So the concentrations, we are using concentrations, so it's concentration of tocotrienols per fresh weight of the kernel, and we don't see so there is a high difference between kernel weight when they are mature, but not when they are fresh. So it's just instead of having starch, they have sugar. So in total, they should have similar weight.
You're saying they have similar dry matter content?
Yes, we haven't tested that, but yes, I believe that.
I don't think that's what you said, is it?
Yeah, it's not.
What? Sorry?
Did you really say they have similar dry weight?
At the
Dry matter at when harvested at the fresh stage.
yes.
I'd be surprised if that wasn't true. I believe that.
Yeah, like we cannot tell the difference between the kernels like visually when they are fresh, but then they are really different when they are mature.
It's a trade-off between wet sugar versus starch.
Exactly.
Mm-hmm.
So they won't have the same dry weight.
You'll measure it. Another question for Matt?
What was the source of the high pigment alleles?
What was the source of the high pigment alleles?
Were they existing within your sweet corn panel, or did you intergress them in?
No, they were existent. So our lines were from public breeding programs, and then we have a lot of variation for the pigment. So we have since like white grain to dark yellow. So we didn't integrate anything so far. So everything was the natural variation in the panel.
Okay, Matt, I think we should. That's very interesting. Excellent. Jin Leung Choi, all the way from Iowa State, will talk about the dirt on soil microbiomes. Please come up.
Thank you. Hi, my name is Jin-Young Choi, who is a postdoc. I work with Adina Hao at the Agriculture Biosystems Engineering at Iowa State University. Today, I'm going to talk about the microbes in the soil. So I know many audiences here are studying plant, but I want to tell microbiome is also very important in the soil. So, example, the plant provide the nutrients to the bacteria to grow, and also bacteria provide the nutrients that plant cannot make by themselves. So they are working together to grow together. But the other hand, there's also not bacteria that take advantage of the plant. They are like a pathogen. So together, bacteria are still very important in the soil and the plant, and especially in the agriculture. But how much do we know about the microbe in the soil? So we used to rely on the isolation when we study bacteria and microbe in the soil. But because of the DNA sequencing technology available, now we are able to sequence everything, all the DNA in the soil. So when we used isolation, that was actually biased. Not all bacteria can be grown under the agar plate. And also that special environment gives us some selection pressure. But DNA sequencing technology, we can collect all DNA, theoretically all DNA in the soil, and we can sequence everything fairly, everything. So compared to the isolation, we have less biased picture using the sequence. But I'm not going to tell we don't need isolation anymore. Actually, eventually I'm going to say isolation is still very important, we need. So, so most importantly, because of the sequencing technology available, we are able to tell how much we know about the soil. About 5 or 10 years ago, when I go to the soil microbial conference, many people talking about we don't know much about the soil microbe. And if you Google soil and question, you will see a lot of picture in the big question mark on the soil. But because of sequencing technology available, now we are able to say how much we know about the soil. Of course, we know many things about the physical and the chemical property, but I'm talking about who is there, what kind of bacteria in the soil. And most importantly, we didn't know how much we don't know. But now we can tell how much we know and how much we don't know. So to answer this question, I used data from the Earth Microbiome project. So in the picture, those that represent the sample in this project. So there is the 14,000 samples in this project. They are over the 16S rRNA sequence, amplicon sequencing, and they take the sample and extract DNA and sequencing under the same protocol, using the same primer, using the same platform. And among 14,000, there are 3,000 soil samples. So the red dots on the map represent where the 3,000 samples from. So it is very widely distributed in most of places in the United States and also worldwide. So I use this 3,000 sample to find what is a global pattern of the microbe in the soil. So let's talk about the soil diversity. We know soil is very diverse, but how much diverse? So what I found is among 3,000 samples, I found 2 million OTU. OTU is the operational taxonomy unit. It is very similar to species, but not really species. But this is the cluster by the 97% identity using 16S ribosomal gene. So when I say 2 million OTU, it's roughly similar enough to understand 2 million species, but not really true. So let me take a guess. So those in here, x-axis is the number of observation among 3,000 samples, and how many samples we found, and the y-axis means how many OTU we found. Let's take a guess what this looks like and what do you want to be like. For example, line number 1, if it looks like number 1, which means actually there are many microbes we share in the many places in the soil. If the line number 3, only few microbes we found commonly, and almost every bacteria are rare. Line number 2 means kind of in between. So the answer looks like this. So I can tell soil is very diverse, very heterogeneous. So most of the OTU we found in the soil is very different from other places. For example, bacteria in the soil in the Iowa, their species, the known isolate from the soil, and that is a graph on the left side. This is also showing the similar pattern with the global pattern, but still many isolates are from the common guy. So our isolation is still a little biased. And also, next question is our known isolate represents how many observed cells in the environment. So the answer is 0.5%. So we, last many decades, we isolate many microbes from the soil. Still, we only have 0.5% among all the environment. So to make together, soil is very heterogeneous and very complex dynamics. So soil is very difficult to study. But so what do we need to, in this different difficult environment, how we study? We need a reference. We need a very good reference. For example, human microbiome is a good example. So human microbiome project is the led by the NIH, I believe, and that is they started from human gut microbiome, which is fairly simpler than compared to the soil environment. And they isolate 3,000 microbes from the human feces, and then they are able to cover most of the population in the human gut microbes. So similar to that matter, we try to collect all the information what kind of microbe we isolate from soil. So I kind of stick to the source of the information, like all the isolate from the soil that we know it is not from other place, and not a very extreme environment, because we don't want to be an outlier. So here is the overview of the reference soil database. This is what I made. And this is a kind of general information how much, what is the general composition of the microbe distribution in the soil. So most of people know proteobacteria is dominant in the soil. It is true. And what is next? Firmicutes and actinobacteria is both second and third highest, and Bacteroidetes. Okay, we have this information. Then what we can use it for? First, we can identify target for the culturing effort, and also we can, after we know who is there, then later we can use this information, what are they doing, and make data analysis easier for soil scientists. So first, identify target for the culturing effort. So actually, I already mentioned about EMP project. So this is the 2 million OTO we found in the Earth microbiome project. And the outer ring, the orange color, represent something we found in the soil reference database. And for example, here in the proteobacteria, proteobacteria is the most studied because this is easy to grow and high population in the soil. So many people are able to isolate proteobacteria, and there's many studies in proteobacteria. So actually, we cover many about the proteobacteria. But still, in here, look at this gap. Even though one of the branch, we totally miss one of the branch in the proteobacteria. So the message here, even though proteobacteria are fairly easy to isolate, we studied many so far, but still we still many are missing in the proteobacteria. How about this? This one, we only have one isolate among these phyla. So this is a really less studied phyla. This one is even worse. We know there is a phyla existed, but this phyla, we don't have any isolate from this phyla. So this is the list. There's a fairly long list, but especially TM6, TM7, those number means we know there is bacteria, but we know nothing about them. We even don't have a name. So if you are looking for, so isolate some cool guy, maybe those are you want to target. So, but so far I say soil is very difficult. We don't know many about it. But I want to say we still have hope. We still did a pretty good job, and we are not too far to reach good. That's what I'm trying to say in this slide. So this graph showing the y-axis is the number of OTU we can observe, and y-axis is the number of coverage. So in this graph, if we observe, isolate over 10,000 OTU, actually we can cover over 70% in the abundance in the soil. So as I said before, human microbiome project have 3,000 OTU to, say, representatives. And 10,000 is only 3 times more. And they did it good job, and why we cannot? I think we can do that. And also 30,000 or 78,000 OTU, we can cover 90% of the microbe in the soil. So it is true most of the OTU are very rare, but we can focus which is the high abundance and the ubiquitous. Then we can actually able to cover many percentage in the soil of microbe. So this is a list of the most wanted of isolate that from the top to bottom showing the most abundance in the soil and also most ubiquitous. So, but which have not been isolated before. So many Vericol microbita and Firmicutes and Eccidobacteria showing here. Well, actually, it makes sense because those are the slow growth bacteria. It's a little difficult to isolate. That's why, understandable, it haven't studied very much. But we know they are present everywhere in the soil and the high amount. So probably they provide very important function in the soil, and we don't know much about it. So probably this could be a good target for our next research. But as I said, these are very difficult to isolate. We need some other strategy other than sticking on the agar plate. So this is one of the possible way to figure out those guys, which is the single cell sorting and genome application. We collaborate with the alumnus from the Bigelow laboratory, and which is the left side panel showing that this is basically a fax and cell sorter. And so in this technology, we are able to sort the cell, sort the single cell in individual well. This application was widely used in the water environment because the water is already, the bacteria and the microbes are already dilute in the water, and this application working very well. But this technology was difficult to use in the soil because the soil microbes are clucking together and a little difficult to separate each other. So, so far, not many people using this technology in the soil, but in Bigelow's lab was able successfully use adding making the standard protocol to handle the soil microbe using this technology. So after the sort the single cell into each well, using the random primer to amplify theoretically almost everywhere in the genome, and then sequencing. Then we are able to sequence something, bacteria and microbes without culture. So those are the 14 list of the genome that we tested and with the single cell technology. So still there are some proteobacteria, but we choose this one and to try to fill the gap, something we haven't isolated in the soil. So there's many Vericol microbita and others, some actinobacteria, something something is novel in the soil isolate. So far I talk about who is there, who is in the soil, and now let's talk about what are they doing. Because, so this is the function that the microbe in the reference soil genome have. And x-axis means the different cluster, the class, and the y-axis the function. And we can see some of the bacteria have, those different bacteria have different function. And why I tell you this, because in the soil, who is there is not enough to find the story from our research because, for example, there's a bacteria A and B. If we use 16S sequencing, we usually cluster define OTU with the 97% identity. Some of the bacteria A and B have the similar 16S rRNA gene, like let me say they are 98% similar each other. But then they will cluster in the same OTU. But it is possible those two bacteria A and B have different function, and we cannot tell this function is from which one. And also it is possible there are two bacteria are different, like they are even different phylum or class or family. But it is possible they share the same function. So not only just figure out who is there, what are they doing is also a very important question. So here's one of the examples that using this soil, reference soil data, is targeting the function. And this research focuses on antibiotic resistant gene and the integrases found in the reference soil database. So here we found that many of the antibiotics resistant genes occur together. They are present together with the integrases. So we are able to guess that integrases and antibiotic resistant gene are something happen together or have a correlation. And also we can use the pathway to analyze the data on our research. Actually, this is a full pathway in the KEGG database, and I overlaid all the functions we found in the soil database. And we found that actually the most of the bacteria in the reference soil database cover most of the pathway in the KEGG database, but not much in the glycan biosynthesis and metabolism of terpenoid or polyketide. And last, we provide a contextual framework of the understanding of soil. So in here, we can, for example, this is done by one of the graduate students in our lab, Gerard. He is interested. We know there's a different type of soil, and I want to know whether there is a different bacteria live in there. And the answer is true. So the x-axis here is showing the different soil type, and y-axis are the bacteria in the soil reference database. And the first column is the molly soil. It is not surprising that molly soil is considered as a fertile soil, and many of them are isolated from the molly soil. And it is very diverse. And compared to body soil, second column is the body soil. It's a different type of soil, and we see the different pattern from the molly soil. So it is true, still some bacteria present everywhere, even though different type of soil, but actually different type of soil have the different membership of the bacteria. Here is another example of using the DNA sequencing technology to our real research. This is a collaboration between our lab and the Indigo industry. And we use the genome to field project data and searching the indigeneity of the isolation. For example, if there is a new isolate, we want to know where are they from, and if it is commonly found in the United States. And you can use this data really appreciate the genome to field project that we can able to say this new isolate is present, and they are originally present in the certain state, and left side showing the presence and absence, and those green and red state means we have a sample collected. So the green means we are able to observe that isolate in those states, and the red means we could not observe it yet. The right side panel showing that how much is there. So this isolate is, looks like highly present in the Illinois, but not much in the Michigan, for example. So those, we are able to provide this kind of information, and this could be very useful in the real world. And I appreciate your attention, and I'm also take any question if you have. Yes.
Two quick questions. Yesterday we saw that micronutrients tend to segregate out to below the surface and then further deep down. I presume you're sampling it just below the surface. Do you have any plans, or have you been sampling as well greater depths to contrast what you're observing?
That is a good question. So many literatures say different bacteria community in different depths of the soil. It is true, but actually the soil sample that I'm showing is doesn't matter which depth, we just collect as much as possible. But they are using the same protocol of the taking sample, so they have all the information, what is a centimeter from the depth and so on. So we are able to figure out what is the depth. But to answer your question, this data have everything of the all depths.
I noticed that you don't have any sampling of Africa, and a lot of companies are interested in ultimately moving there and getting ecotypes of what's there so that they can actually ultimately go to Africa. Do you have any plans in the future of sampling any soils in Africa?
So nice thing about the NGS technology, actually personally, I don't have a plan to trip to Africa, take a sample at this moment, but we are able to collaborate with other people. And also, if somebody, if anybody sequence their DNA, most of the time they post their sequence in the NCBI, and everybody can be used for free. So there are many more data on the publicly available. So we can use other people's data for our research, and especially other countries we don't travel to.
So can you give us a better sense of what's going to happen with the genomes to field samples?
So I visioning that this project is already provide very good advantage and to the many research and industry. But if we expand to the cover the most of the state in the United States, and
I know with the existing samples, the materials you have now, can you tell us what's going to happen?
So your case.
And I think they're asking about the indigo samples that were sent to Indigo. Do you know what's going to happen to the samples in Indigo? What's the next steps for them?
I believe the things about the indigo is maybe confidential to talk about public.
The data has, so they've been extracted and analyzed, and that information is available to Genomes to field as well as any, we've got frozen samples as well.
So we can get access to copies of that data?
Sure. That was the whole idea.
Yeah.
I mean, this is public.
Sure.
We're certainly interested in doing it again.
Okay. So even same samples, same sites, but different years will be interesting too.
Yeah. Yeah, and we may also want to think about some sites doing a little bit more of a course through the year. We will be doing that at some of our sites. A lot of this is really about understanding the diversity that's out there,
science paper also looking at Earth microbiome data that also drew up a most wanted list of like microbes we most need to fill in our gaps. Have you had a chance to compare your list with their list and figure out how much overlap there is?
So actually, I haven't found that particular paper you mentioned, but I will be happy to compare with my list.
Okay, I think we better move on. Thank you, Jinzhen. Liji Wang, kids. Okay. Liji Wang from Iowa State University will be talking about improved strategies for genomic selection.
Okay, good morning. I'm going to start by thanking the organizers of this workshop. They have been doing an excellent job, and this has been a wonderful learning experience for me. My name is Liji Wang. I'm an associate professor from the Department of Industrial and Systems Engineering at Iowa State University. I'm going to present a new algorithm for genomic selection. I call this look-ahead selection algorithm. So let me start. Let me start with a problem definition. Conventionally, breeders make selection decisions based on the phenotype. Suppose we try to breed for taller plants, and these plants have different heights. So what do we do? How do we select two individuals to make a cross? We are going to select the tallest two to make a cross. And this is based on the assumption that taller parents are going to produce taller children, which is very intuitive. The invention of genotyping technology enabled people to make selection decisions based on the genotype information. So people can collect the genotype information as early as the plants are still seeds. And then based on the genotype information, they can do genomic prediction and estimate the effects of those alleles. And then if you add the effects of all the alleles, you get some breeding values, such as the genetic estimated breeding value. And then that could be considered as some type of prediction of the plant height. So what they do is very similar to phenotypic selection, they rank the predicted plant height and select those that are predicted to be the tallest when they grow up. Very recently, there's a new research direction looking at new and improved algorithms for genomic selection. So we're saying if there is no genotype information, the only thing we can observe is the phenotype. It makes sense to assume that taller parents produce taller children. But now we have mountains of genotype data. We may be able to do a better job in achieving higher genetic gain by using more advanced algorithms. So let me briefly review what has been proposed in the literature, which is not very many. So this is the conventional genomic selection algorithm. So you see the GEBV, genetic estimated breeding value, is defined by the summation of beta and G. G is the allele, and the beta is the effect of that allele. So the GEBV is estimating the genetic value of the individual breeding parents. And we are going to rank them and select the individuals with the highest GEBV and remove those with lower GEBVs. So this approach was proposed in 2001 and has been widely adopted. And people have shown that this approach is very effective in achieving higher genetic gains, but it's for short-term genetic gains. A limitation of this approach is that it tends to lose genetic diversity and long-term growth potential. But this approach was so popular that for more than a decade, people have been using this approach or some minor variations of this approach. Not until 2015, we saw another very different approach. It's called OHV, optimal haploid value. So the OHV is defined as as this equation. So the idea is that it's defining some haplotype blocks, and for each block, it's comparing the pair of chromosomes and selecting the better haplotype block and adding all those better blocks. That's how we define the OHV value. So what this OHV tells us is that for a given individual, if you solve it, the OHV is the best possible progeny you are going to get by self-pollinating this one individual. But assuming that there's no recombination within the haplotype blocks, but there could be recombination between them. So this approach is a very good improvement over the GBV approach because it represents a shift of selection criteria from selecting the individuals with the highest genetic achievement themselves to selecting the individuals with the highest potential to pass on the best genes to their future generations. But this approach also has some limitations. It is still a truncation selection, meaning we are ranking the individuals based on the OHV. Okay, and this is assuming that the contributions of an individual is separable and additive. Last year, my colleagues and I published a paper, and we proposed a different genomic selection approach. The co-authors are Professor Pat Schnabel and Guiping Hu, who is also an engineering faculty member, and myself, and two of our students who, one is Aaron, who is here, and the other Matt, he is also an engineer. So this OPV value is defined in a different way. So instead of defining a value for an individual, the OPV is defined for a group of individuals. It is defined in a similar way as OHV. We are also defining haplotype blocks. But for each block, we are looking at the entire group of individuals and see which one has the largest block. And then we add up all the haplotype blocks. So what this OPV tells us is that if you cross this group of selected individuals, what's the best possible progeny you could get? Okay, assuming you have infinite amount of time, assuming you have infinite amount of resources. Okay, and this approach represents another important shift of selection strategy, shifting from selecting based on the individual value to a group value, because the contribution of an individual, we believe, cannot be estimated separately. You have to look at the complementarity of all the individuals in this group. But there is also some limitations. This approach is making the assumption that we have infinite amount of time and resources, and it's very computationally expensive. Now let me talk about the ways that we can further improve these approaches. So one direction of improvement is considering the time constraint. Just look at these two approaches. Approach one is achieving genetic gains slowly, but it goes up higher. Approach two goes up quickly, but also plateaus very early. So which approach is better if we want to achieve higher genetic gain? Of course, it depends on the time. If you have a shorter timeline, T1, I would prefer approach two. But if you have a longer timeline, approach one is better. If you look at the existing genomic selection approaches, they don't care about your timeline. But we are saying we should consider how much time you have, what's the project duration. And the second direction of improvement is considering resource constraints. So consider the two crosses. Cross one is between two shorter parents. Cross two is between two taller parents. But for cross one, the two parents are very different in terms of their desirable alleles, okay? Because they are so different, they're complementary. Their progeny have a wide distribution. And for cross two, suppose that they are very similar in terms of their genotype. So their progeny have a very narrow distribution. So which cross is better? This also depends on the resources we have. If we can only produce one progeny from each cross, I'm going to bet the cross 2 is going to be better because it has a higher expected value. But if we have a large amount of resources, we produce thousands of progeny, we are going to believe that cross 1, the best progeny from cross 1 is going to outperform the best progeny from cross 2 because cross 1 has a bigger tail. The third direction for improvement is considering mating strategy. This is an experiment using real data. Suppose we have selected 4 individuals, A, B, C, and D. But how do you pair them up? If you cross A and B and cross C and D, this is what you get on the top. At the bottom, if you cross A and C, cross B and D, that's what you have. So you see the results are very different depending on how you pair them up, even if you selected the same set of individuals. But if you look at the existing selection algorithms, they do not tell you how to pair them up. So a lot of times, people just randomly pair them up. But we're saying if something makes a difference, we should optimize the decision rather than leaving it to a random toss of coin. So let me present the new approach. The objective of this so-called look-ahead selection approach is to maximize the probability that we are going to produce an outstanding progeny by crossing the selected pairs of individuals by the project deadline and given the resource constraints. So notice that we are considering a lot of things. We consider the project deadline. If you have a different deadline, the approach, the selections will be different. If you have a larger amount of resources or smaller resources, the selections will be different. And we also select the individuals in pairs, so they're already paired up in the optimal way. And we maximize the probability of creating an outstanding progeny. The method we use is called Monte Carlo simulation based on conditional probability calculation using recombination frequencies. There's a lot of equations. I won't show them, but I will show you a more better looking picture on the next slide. The input for the algorithm is some data, genotype data, recombination frequencies, the timeline and your resources constraints, and the output, this is an example of the output. We have a matrix that has 4 rows. 4 rows means we are going to make 4 crosses, but this is a decision variable. It could decide to make 3 crosses or 17 crosses. That's a decision to be made. The first column and the second column are telling you which 2 are the breeding parents for each cross. So the first one is you cross 12 with 16, and with 69. Second cross is you cross 247 with the same 69. So the selected individuals are already paired up. The third number is to tell you how many progeny you should produce from each cross. Okay, so this picture shows the idea of the Monte Carlo simulation. Now, starting from this given from generation small t, that's the selected pairs. They're already paired up. And then in the next generation, they are going to produce a random progeny with some potential recombination events. And then the capital T is the terminal generation. We know the project deadline. And then we are going to simulate what the final generation, what the distribution is going to look like using some advanced probability calculation algorithms. And then we can estimate what's the probability that in the final generation, you will see an outstanding progeny. And then we are going to do an optimization. How do you optimize the selected individuals and the mating strategy in generation small t so that in the final generation, you have the largest probability of producing an outstanding progeny. So let me show you a case study. The case study, we collected some data from Pat Schnabel's lab. It's 369 inbred lines of mates data. Each has 1.4 million SNPs. And we also have the recombination frequency data, an estimated effect of each allele. The initial population, we just randomly select 200 lines out of the 369 as the initial population, and suppose our duration is 10 generations. The criteria is that we are going to compare the best progeny in the 10th generation, but we don't compare it once. We do the simulation, we repeat it thousands of times and look at the cumulative distribution function. So this is the result of one simulation run for the conventional genomic selection approach. Horizontal line is the timeline, vertical line is the GBV. Consider this as the height of the plants. So in generation 0, you see the dark blue bars, they are giving you the histogram of the initial population. And then we are going to select the top, the tallest lines, randomly make some crosses, and then generate the next generation. And again, we select the top lines, make some crosses, select and produce the next generation. So you can see the average GBV is increasing over time, but very quickly there is no diversity in terms of their height. So you see this is very narrow, but it's also 200 individuals. They're just very similar in terms of their GBV. On the next slide, I'm going to shrink this figure. I'm going to change the ratio, make it very narrow so that you can compare it with some other approaches. So this is, on the left-hand side, the conventional genomic selection is the same figure as you saw on the last slide. It was achieving a genetic gain of 59. Using the OHV, optimal haploid value selection, it was able to achieve a genetic gain of 64. OPV, 68, and look-ahead selection, 69. And very nice thing about the look-ahead selection is that you can see there's very nice diversity in terms of their plant heights throughout the entire 10-generation period, rather than the conventional genomic selection, where very quickly there's no diversity. But I also want to emphasize that this is only one random sample run. Okay, so if you run the sample at a different time, you are going to get a different number. I have run the simulation for thousands of times, and I was trying to find one case that the look-ahead selection was doing very poorly, and I was able to find one. It's very rare, but I found it. So I'm not trying to pretend it's always better. So this is a case where the look-ahead selection was underperforming. It's only achieving 59, where others are being better. But again, this is a very rare case in thousands of simulation runs. So now let's compare the cumulative distribution function, which gives you a better idea of their performance in thousands of simulation runs. The horizontal line is the genetic gain. The higher, the better. Vertical line is the percentile. Okay? So if you look at the white curve, this is the conventional genomic selection approach. The 0% is the worst case in the thousands of runs. 100% is the best case. Okay? And 50% is the medium case, or close to the average case. So that's the performance of the conventional genomic selection. OHV, it is doing better because it is moving to the right-hand side of the curve. It's achieving a higher genetic gain on almost every percentile. OPV is also better, and this green curve is the look-ahead selection. So you can see it is outperforming all the existing selection approaches in the literature. And then I also do a sensitivity analysis. So far, I'm assuming that we are able to do an excellent job in predicting, in doing genomic prediction. We know the true effects of each allele. What if we don't? Okay? So I did another experiment. So I assume that the data we have is ground truth. It's the ground truth effect of the alleles, which we don't know. We only use the ground truth to evaluate the performance of the algorithm. But the beta hat we are using in the selection algorithm has some errors. Okay? So the errors, I just arbitrarily add some errors. So for half of the SNPs, I'm assuming that they are correct. They are equal to the truth beta. For a quarter of the SNPs, I'm assuming that they are overestimating the beta. So it's twice of the true beta. For the other quarter, I'm assuming they're underestimating. They're only half of the true value. And see what happens. Okay, if we use the beta hat instead of beta for the genomic selection algorithms. That's the result. So you see many curves. Let's look at the white curve. There's a thicker curve that's assuming that we have the perfect information on the beta. Okay? The thinner curve is the imperfect estimation of the allele effects. So you can see because of the error terms in the beta, the curve is moving to the left-hand side slightly. It is a little bit worse than if you know the perfect information. And for all these cases, the thinner line is always moving slightly to the left. I want to point out two observations. One is that the OHV and OPV, they are more sensitive to the errors in the prediction results. So if you have some errors, the thinner curves move further away from the thicker curve. On the other hand, the conventional genomic selection and look ahead, they are more robust in terms of the errors in the beta hat estimation. Second thing I want to point out is where we are right now. We are on this thinner white curve because most of people I've talked to, they are using the conventional genomic selection approach. And we don't have the perfect information on the beta. That's where we are. And a lot of people are trying to improve the prediction accuracy. But the point I'm making is that even if you make it perfect, if you make perfect prediction, you are going to move from the thinner white curve to the thicker white curve. It doesn't improve that much. But if you adopt more efficient selection algorithms, such as OHV, OPV, or look-ahead selection, you are going to make much higher genetic gain improvement. I want to acknowledge my collaborators and supporters. This work is a joint work with my collaborators, Professor Pat Schnabel and Guiping Hu, and Saba is the student that has been providing some help on this look-ahead selection. And this was supported by the USDA NIFA grant. I'm also very gratefully and proudly supported by the Plant Sciences Institute at Iowa State University. Thank you for your attention. I'm open to questions.
Got a question in your approach. So you compared your various ones. Conventional selection, genomic selection went up very fast and then plateaued, and the others were sort of a trade-off. You got better longer term, but it took a while. And so you're saying that they're more efficient. I'd actually argue the conventional is more efficient because you get more gain per unit time, at which point in the real breeding program, I assume you stop trying to breed within the same circle of germplasm, you bring outside stuff in. So how does this whole thing change if you don't use a closed breeding pool? It's an open breeding pool where you can bring in outside germplasm at various points to try to enrich it once you've depleted it.
Okay, let me repeat the question. The question is, the argument is that the approach 2, which is the conventional genomic selection approach, is actually a good approach because it can achieve short-term gains very quickly. And then if you bring in outside varieties, you can achieve another short-term gains. Is that the question?
Yeah, that's basically
So, but my response is that suppose you're not expecting outside new varieties, or you're assuming that you have outside new varieties already in generation 0, okay? We are going to achieve genetic gains by not only achieving only part of the potential, but full potential as much as possible. Okay, so if we bring in all the varieties that we have, okay, we do not necessarily want to just achieve 80% or let's say 40% of this very quickly without any potential for future improvement. because genetic improvement is not a short-term deal, it is a long-term potential. We do not want to lose the genetic diversity and long-term potential, which you may not find anywhere else.
So kind of going off that, then would you say, well, because right now people talk a lot about how there's less and less genetic diversity in the germ-breeding plasm, the germplasm. So you're saying this is a better approach because it'll make full use of the limited genetic diversity we have?
That's right. It is using full use of the genetic diversity we have, but also it's making a trade-off between how much time you have and how much resources we have. Yes?
Great presentation. Can you repeat the major advancements from the OPV to look ahead? Besides the time constraint, seems to you can do that in OPV. So what is that thing you did that make this looking ahead better than OPV?
Okay, question is the improvements of the look ahead over the OPV. OPV is a very good, a very efficient algorithm, okay? But the limitation is that it's assuming that you have infinite amount of time and infinite amount of resources to achieve the best possible progeny, okay? But the look ahead selection is saying we are going to take into consideration how much time you have and how much resources you have, okay? If you change the time duration, if you change the resources, it's going to change the selection accordingly. Because of that, it's able to achieve a higher genetic gain based on the resources and time constraints we have. Yes, James.
You're explicitly modeling resource constraints,
Mm-hmm.
and then it gives you a strategy based on that. I mean, do you have one pool of resources to say, you know, for either I can make this many crosses, or if I have fewer crosses, I can advance more progeny? Or do I have to say I can make 20 crosses and I can advance 2,000 progeny? I mean, do you consider
Okay.
between resources going in one step?
Okay, I think the question is regarding what do I mean by resource constraints.
Yeah.
The assumption is this. Let's say we have $30,000. That's the fixed budget. Every time you make a cross, it costs you, let's say, $10. Every time you produce, for every progeny you produce, it costs you $5. Okay? You can decide how many crosses you make. You can decide how many progeny you produce. Just don't go over the budget. So you have to decide how do I allocate my budget to different generations, to different crosses. Yes, sir.
So we know the relationship between, and I may have missed this, we know the relationship between genotype and phenotype sort of decreases over generations. So how often were you updating the phenotype-genotype relationship and then retesting that in your experiment?
Okay, question is regarding the genotype-phenotype relationship. In this study, I'm not considering the phenotype equation. I'm assuming that the genotype is the main contributor to the phenotype, so only looking at the genotypic effect. I mean, assuming that you know the true effect of each allele, just add up all the effects of the alleles, that will be the genetic gain.
So you're not updating the model for us?
No, I'm not updating the model. So, or I'm assuming that I already have the ground truth of the effects. The genomic prediction was already perfect. Yes, sir.
In the simulation data, did you actually use any inputs that were from the 200 inbreds? You said you have 200 lines that were randomly selected.
The inputs we use from the 200 lines were the genotype information, the recombination frequency information, and the assumed allele effect information.
The assumed allele effect information for the trait plant type?
That's right, that's right. So in two cases, in the first case, we assume we know the ground truth, or we assume the genomic prediction was perfect. In the second case, there was some errors in our estimated beta.
So my follow-up question is, do you think that the granularity of trait values matters, and that the simulation outcomes across the four methods would be the same if you simulated for yield? rather than for plant height?
Oh, I think that's an excellent question regarding the granularity of the markers. So in this case, we are using 1.4 million SNPs. I think my next step would be considering what if we only use 10,000 of them. So is that going to make a big difference or not?
I think the motivation for that's interesting, but I think the motivation for my question is surrounding the trait complexity of plant height versus the trait complexity of yield. And that the granularity of the individual genetic contributions may be different.
Yeah, that might be some good questions for further research. Yes, ma'am. Okay, question is regarding evaluation for each generation. In the simulation, we can get the evaluation based on the phenotype information multiplied by the allele effects. But I do not know how this translates to real breeding process. If you don't see the phenotype, you may not get the feedback unless you do the genotyping.
To me, it would be of interest to include something that was, genomic selection plus any consideration of an informed cross.
I think the question is regarding genetic diversity.
It was sort of more of a comment, I guess, than a question.
Okay, okay. I think the look-ahead selection approach, although it doesn't specifically maximize diversity, it is actually doing an excellent job in keeping the diversity. If you just look at the curve, it's the diversity in terms of GBV, I think represents the diversity in the genotype information.
No, I agree. I'm just saying your left-hand panel, I think to have a fair comparison.
Oh, I see. Yeah. So you're saying this is not the actual practice?
Yeah.
Okay, yeah. But that's a simplified version of the actual practice.
Yeah, but you can imagine you would begin in each case with all the diversity that exists in the world.
Yeah.
And then that would, instead of bringing in this notion of adding diversity sequentially, let's imagine you had access to everything, and then you ran this prediction. His method would tell you how to take advantage of the universe of diversity.
Right, it doesn't mean to add anything in. I mean, given what you would start with, if crosses you make are going to be informed not only by what's the best thing, but also what's going to maintain my selection for the next generation.
But if you think about this example as one F2 population of 200 that the breeder's doing, then you have the variation from the 200 population as a result. And so it depends on, I think this is the whole breeding program, or if this is one of 100 F2.
Yeah. Are you planning an experiment where you do this one and this one?
Currently, I don't have a plan, but I'm always looking for collaborators who is able to test it out.
Where's this novel guy with the 200 lines?
It's an expensive experiment.
Oh, is it?
It is.
Yeah.
We proposed it. It's expensive.
I mean, it would be fascinating if it played out.
Yes.
Like that.
So I have something quick to add. The Atlas project, led by Randy Weiser, characterized the shifting allele frequencies over the 10 cycles of Hallauer's Toussaint. And you can clearly see that in early cycles of selection, certain alleles get fixed, and they appear to then capacitate the fixation of additional loci, and then again and again over 10 cycles. Really a lot like what you see in some of these panels of it going up and like a little intermediate plateau and then going up again and again. I think it's a really interesting finding. His manuscript is under review.
I get the impression that Ligia is an excellent teacher. What do you think?
Thank you.
Thank you. So, next we shift gears now, where we go to a sort of an important denouement or summary now, right?
Yes.
Pat, you're taking the lead, right? It says Carolyn should.
Carolyn is going to take the lead, and all I'm going to say is Carolyn's going to take the lead, but we're going to take a 10-minute break. So go take a break. Are we meeting back here?
Mm-hmm. Yep.
We're meeting back here, and Carolyn is going to do her magic.
Do you want me to
Yeah, if you
put
can
it on
put it
here
up so
quick?
it's
Yep.
when we get.
And then we're going to.